All right, good afternoon. How was everyone? Breathing. That's a good start. Breathing is good. Respiratory skeletal muscle activity. We'll talk about skeletal muscles today. Good segue. I like that. What's happening on Monday? Exam 1. Where is it? Right here. What's the format? I got you. What's the format? 50 questions, multiple choice. It's a test packet, paper handout with a Scantron, a number 2 pencil, like the old school way. Okay? On your Canvas shell, there's like an assignment that says Exam 1. I just put that there so it shows up in your calendar feed. So some of you are thinking like, oh, is it going to be online? No, it's just so you see it so you don't forget, okay? I'm not sure how you forget because we've said it so many times, but also on Monday, I'm going to have expanded office hours. Expanded office hours from 1 to 3 before the exam. This is like your last minute. I want a few questions answered. I'll be on Zoom or I'll be in person in my office in Wetah. Okay? There may be some pesky pathology students at 1 o'clock, but you guys will probably outnumber them. All right? We're backing up. Sunday, 3 to 4 in the afternoon. Is that right, Amara? Sunday, 3 to 4, Wetah, room 242. That's right? That's your last review session. Sunday, 3 to 4 p.m., Wetah, 242. The Sunday one probably should be here, just because you have a key to this room, and on Sundays the university's locked. Scratch that. This room, Sunday, 3 to 4, because that key you have will get you through that door. Thursday, 6 to 7, Wetah, 242. Thursday, 5 to 6, Science Annex, 122. Wednesday, well, that already happened, 11 to 12, Science Annex, 122. So you missed that one, okay? There's another participation opportunity. If you go to one of those sessions, you'll have a participation opportunity worth 5 points, okay? Kind of encouraging you to study and stay on top of stuff. There is an extra session by another student of mine. He's actually in my research lab, and he's off. He's TAing for another 201 class, but he said that you all are invited. His name is Parker Bogus. He's solid. This Friday, September 13th, 1 to 2 in this room, Bio 265. That's Friday, 1 to 2. He won't have the participation code. So you got to go to one of the other ones for the participation code. And the Sunday one, we're going to try to have it on Zoom, right? And recorded in case you miss it. Questions? Yeah, all of these are on Canvas, but absolutely. Thursday, 5 to 6, Science Annex 122. Thursday, 5 to 6, Science Annex 122. Did you need any others? Okay, all right. We have study guides that are published under resources in Canvas. I believe also Eliana posted those on Canvas through an announcement, so you probably got those multiple different ways. How should you look at the study guides? How should you treat the study guides? As a resource, as a reinforcement resource, not as a replacement for lecture. Where do I write my questions from? From lectures, from my lecture notes, from the lecture slides. So a couple things, housekeeping things. I'm updating the setup for AV lectures. I got new microphones and I've got a new iPad, and I even bought a new tripod because the other one was broken. I was like duct taping it. So I'm still kind of getting used to it, and I think the wireless mics are really nice, but they don't have as long of a battery life, so I got to switch out halfway. So on YouTube, for the last 2 lectures, the last few minutes, we lost audio. So I just clipped it, and then I grabbed a clip and shortened it from the spring semester that was over those slides. And I watched them to make sure that it was still relevant, and I said kind of some of the same stuff. Okay, so that's why you're going to see F of 24, which is fall of 24, 1 of 2, and then you'll see spring of 24, 2 of 2, as a file that's like 2 minutes long or 5 minutes long or whatever the number is. Hopefully today it'll go nice and smooth, and you'll just have one audio video file of fall of 24. This is what happens when you got the one-man show up here. I actually do want to go through a couple of things about the exam, just to kind of orient you. I would walk through maybe the notes that you have, rewatch the lecture slides, and organize it. Like, you could organize this based upon the fact that, okay, we have 50 questions. We've got an introduction, cell biology, chemistry, and histology and tissues. So we have 50 questions, and we have like 4 topics. So that's like roughly what? 12-ish, 12 to 14 questions per topic. So maybe you're looking at your lecture slides and you're saying, okay, what are the possible questions that you might ask on molecules of life, the chemistry stuff? Stuff like pH, right? Denaturing proteins. What's a disaccharide? What's a monosaccharide? Some definitions. It's pretty easy to get to 12 to 14 questions pretty fast, right? But for exam 1, it should be pretty straightforward because you have 50 questions. You're dividing it up amongst about 4 different topics. Does that help? Just to guide you on maybe what to look at. We talked about in one of the early lectures about body position, right? Whether it's pronated or supinated or medial versus lateral. You remember all that? We talked about, I went really quick and I kind of said, I want you to look at these because it's going to help you in lab. That's game on. I also said in lecture what's game on are organelles and what their functions are. Like, what does the nucleus do? And those are some things that you probably reinforced in SI session on the organization of nucleus all the way to the cell membrane. In the early lecture, we talked about like how to organize the smallest to the most complex, right? Structures. Subatomic all the way to the organism. You remember all this? Periodic table, we did a little exercise. That was an online lecture because I was out of town. But I asked you about how many protons or electrons. Do you remember that in the lecture? I would be familiar with how to do that. If you haven't picked up on it, I'm going through my notes and giving you some hints. Okay. I want to... Don't worry, we'll have time to get through all of this. I just want to reinforce a couple of pictures. What was this image that I showed? Skin, integument. What's that top part up here? Epithelium. Okay, great. We also refer to this as epidermis. You're going to get this like next week in lab and next week with us in lecture. We talked about when you grab a sample of skin, what color is it after you process it, before you stain it? Mostly clear. Thank you for mostly clear. I like that. Giving little qualifiers like that, like mostly or often, typically, is valuable. It's usually never and always are dangerous terms. Does that make sense? Okay. What about these brown punctate cells? What did I say those were? Melanin. Okay. All right. Well, you guys were actually listening. Dang. Impressive. Well, I'm in the same I'm in the same. I wanted to go with one more. Oh, this picture. Yeah, okay. This picture. Somebody asked after class. That's why this is coming up. This lower right picture, what is that? What did I say that was? It's a blood vessel. Thank you. And the brown stained nuclei are what kind of cell? Squamous. Are they stratified or simple? Simple. Weird color. I've actually, other than this picture, I've never seen a histology slide of vasculature that's green. Okay? All right. Any other questions that I could clarify before we dive in today? About the exam, what to expect. You should have plenty of time. 50 questions, an hour and 15. There will be pictures on the screen. Probably 5, approximately. Maybe exactly.
With transitional epithelium, is there like any very specific feature that can help us identify that?
With transitional epithelium, is there any specific feature that can help you identify that? Let's show this slide of transitional. Remember what I also said was, hey, if you're going to see histology, you're going to have seen it before, right? It'll be like an image that's familiar to you out of the lecture. So I would either have to give you some clues as to like where it is or what it's doing functionally, or I could show you like a before and an after, like filled versus empty or stretched versus unstretched. Okay? Because you really move from an empty cuboidal to a full where these are starting to stretch out and flatten like squamous. So giving one snapshot in time would be really difficult to discriminate. Okay. Fair question. Okay. All right, let's get going on today's. We just have to finish out a little bit of material. Talked about, let's see, I got all those. Oh, you're going to want to stick around to the end. Got one more reminder to myself. Where are pencils made? Pennsylvania. Well done. Well done. Well, I was going to make a debate joke, but I'll leave it alone. Okay. Why did the skeleton ride the roller coaster? Why did the skeleton continue to ride the roller coaster? Excuse me. It found it humorous. It's good. It found it humorous. I like that. I might have to write that down. But also didn't have the guts to quit. But I like the alternative answer. What do you get when you cross a tornado and a cow? A milkshake. Well done. Well done. I personally like that one a lot. All right. Good, clean fun. Okay. So we're in connective tissue. What does connective tissue do? It connects stuff. Well done. Put stuff together. There's some other lists of things that it can do, right? Remember that? Protection. Absorption. What's that? Like shock absorption. I can't hear that. Say that one more louder. Transportation. Like in the case of like blood, for example. Okay, so knowing the functions of epithelial tissue, the functions of connective tissue. Which one is cell rich in matrix poor? Epithelial tissue. Which one is matrix rich and cell poor? Connective tissue. Does everybody understand what I mean by that? Like, that's not nice to call somebody poor, right? I'm talking about whether there's lots of cells or very little cells. So I want to make sure when I use the lingo, sometimes people are, confused, and I want you to feel comfortable. A small class like this, you should raise your hand. Ask the question. That's the beauty of having a smaller class. Okay, connective tissue. Connective tissue proper. this large group, there's loose and dense, just like it sounds. This is where you are in lab. What we're covering some overlapping stuff this week. It should be very convenient for you. We'll get a little out of sequence and then we'll kind of come back together, but this week is really nice. For exam 1, like we're talking about stuff that is apropos for lab and lecture. So all but bone, blood, and cartilage in this category are made up of these fibroblasts or these fibrocells. And blood, bone, and cartilage have their own cell nomenclature that we're going to talk about. So if we look at loose connective tissue, the first category is areolar connective tissue. Areolar connective tissue, that's how you pronounce that. We looked at a slide already once in Monday's lecture, but this is a loose arrangement of fibrous proteins. We saw reticular, collagen, and elastin, if you recall. And it has a high gel component. If I say it has a high gel component, what does that tell you? What does that hint to you if it's got a lot of gel in it? What's that? Fewer cells, I would agree. Extracellular matrix has fewer cells. It's open. Can you tell me anything about maybe a proteoglycan or what it might attract? Attracts positive ions known as sodium, and then what follows sodium? Water. So if it has a lot of gel component, it's referring to that proteoglycan. That means it's pretty hydrated. It's very moist. It's under the epithelium, under the skin, this areolar connective tissue. It's around blood vessels, a site for diffusion, and there's a loose open matrix for leukocytes to move. Those are white blood cells. Second type of loose connective tissue, adipose connective tissue. It's a matrix. It's an areolar-like matrix. And these adipocytes is another name for a fat cell, adipocyte. This fat cell are derived from fibroblasts. They don't divide. They come from fibroblasts. They differentiate into fat cells. There's copious amounts of subcutaneous fat in humans. Lots of fat around our organs. This connective tissue to kind of hold things together and provide insulation and cushioning. Our organs are critical structures. Like remember the Amazon package example? Like we want to package it with the bubble wrap. Think of adipose tissue as like bubble wrap inside. It stores those neutral fats. We talked about the energy storage capability of fat. It's very efficient. It's lightweight. No water is required. You can move fat into aerobic respiration and generate ATP. Fat has a very rich blood supply. Now, of course, in our country, bless you, we have procedures where you can remove fat, right? Unwanted fat. And you can do this surgically through a vacuum suction known as liposuction. And one of the biggest risk factors for liposuction is hemorrhage because of this principle, that it has a tremendous amount of vasculature. What were two structures that were poorly vascularized or relatively avascular? Tendons and ligaments. We'll get back to those as connective tissue. They're also very rich in stem cells, fat. So we actually do fat transplants now clinically. We'll harvest fat, liposuction fat. We'll isolate the patient's own stem cell and we'll deliver it therapeutically back to the patient. It's pretty crazy. There's companies that actually store fat-derived stem cells, adipose-derived stem cells, and they'll store them for you. They'll bank them for you. You pay a fee. Okay. Next type of loose connective tissue, reticular connective tissue. It's areolar-like, a fine meshwork of collagen fibers. And it operates like a sieve. So you'll find this in a lot of lymph tissue, like lymph nodes and the spleen, where like a like a filter, it's going to trap lymphocytes that are immune cells. Now we're shifting gears from loose connective tissue. We had three types: areolar, adipose, and reticular. Now we're going to shift to dense connective tissue. So if you look at the architecture, the structure of this slide versus these previous slides, there's less openness to them. The tissue is really tightly packed. That's why it's called dense connective tissue. Question? The word areolar means like open, airy, not dense, not compact. Lots of open space. That's what that root word areolar means. So areolar-like is it's kind of like a matrix, or has a lot of porosity to it, like a sponge. Does that help? Yep. So this tissue, often in lab, you'll mix this up for smooth muscle. But smooth muscle has lots of nuclei. This has very few nuclei. So that's the big difference. We'll get to smooth muscle later. But dense regular connective tissue doesn't have quite the vascularization as loose connective tissue. And it resists stretching in one direction, one plane. So imagine it's like sheets. Okay, you layer a bunch of sheets together, paper, and you glue them together. And then you want to pull on them in this direction, right? They're very, very strong. But if you pull on them in this direction, they're very weak. So they're unidirectionally strong. Dense, regular connective tissue. Regular means that they're all oriented in the same direction. Like a lot of structure, architecture, anatomical terminology in the name to give you clues on what it looks like. And now that structure dictates how it performs. That's the physiology, its function, right? So tendons and ligaments are organized as dense, regular connective tissue. So they're strong in one direction. But the moment you move them in the other direction, or in a sideways direction, that anterior cruciate ligament, that kind of the anterior and the posterior cruciate ligament kind of make a cross. That word means cross or crucifix. So anterior crucifix ligaments are the ones that kind of cross each other. So they're strong in these directions, but they're weak in the lateral direction. Does that make sense? That's why they tear. So I don't know. It's a great question. I don't have the answer. Why would it be designed like that? Why not make it irregular connective tissue? We'll get there, where it's strong in all directions. But this has a high collagen content. It's very strong, but it's unidirectionally strong. Does that make sense why? This is irregular, the kind that I'd rather have tendons and ligaments, because then we would put orthopedic surgeons out of business, right? Which is bad for them, but good for patients. So do you see the difference in the planes of the collagen and how they're organized? This is organized very parallel. This one is swirly and all over the place. Dense, irregular connective tissue. Lots of collagen, but it allows for force or tension in many different directions or many different planes. So all of the remaining connective tissues that we're going to look at actually have what we call lacunae, or structures where cells will live. The only one and the only exception is blood, so we'll get to that one in a minute. But now we're moving into cartilage. So I say no blood supply. Some of you might argue that's fine. On the exam, if you see no blood supply, don't get confused. I'm not trying to trick you. I'll try to make sure it says limited or relatively unvascularized or relatively non-vascularized. But just know, like if you ever were in an office, a surgical office, and you said that cartilage is avascular, someone might argue with you, right? So don't get confused by that. I'm not going to try to trick you. And if you feel like you're getting tricked in the exam, just raise your hand and I'll guide you through the question. Okay? So no, we're in connective tissue, but we've moved out of loose and dense. Now we're just into other types of connective tissue. Loose and dense are the main categories. And then cartilage, obviously, there's not as much cartilage in the body. So this has a perichondrium. This is a word, peri meaning surrounding, like perimeter, like the surrounding perimeter. The outside of the cartilage has this perichondrium, which is a layer of dense, irregular connective tissue that you just asked about, and it surrounds the developing bone. Because what we'll learn about later in the semester is bone actually comes from cartilage. First you lay down a cartilage template, and then you ossify it, or you make it hard. And then the ends you keep soft as cartilage, so you have some cushioning. So this perichondrium, I keep wanting to say pericardium because you have a pericardium around the heart. Perichondrium is surrounding the cartilage, and it's that interface between the bone and the cartilage. And on this slide right here, these cells are in this little opening, which is the lacunae, or a little cave that the cells live in. Bone has the same architecture that you'll see here in a second. So we have different types of cartilage. We've got hyaline cartilage, first up. We'll talk about elastic and fibrocartilage next. But hyaline cartilage is the one that I kept showing you on Monday, all these different pictures. Like these pictures were on as hyaline cartilage. It's typically gristly. It's the gristle that's found a lot of times in your meats. Sorry if you're a vegetarian, because you're like, that's why I'm a vegetarian. I don't like the gristle. It's the most abundant type of cartilage. It's made up 40% of collagen. You find it in the trachea, which is basically the windpipe, or the tube that moves air to the lungs. The nose tip, the tip of your nose, costal cartilage between the ribs, and the end of long bones. And there's a nice picture here of a lacunae and a chondrocyte inside of it. The chondrocyte is what makes the cartilage. Next up is elastic cartilage. There's more elastin in elastic cartilage, amazingly, right? External ear, the epiglottis, which is in the back of the throat and actually helps decide if something's going to down the esophagus or down the trachea. These red arrows are actually pointing to these dark, squiggly black lines, which is the elastin that's being stained. Most elastin stains have elastin being a dark, black, squiggly line. That's what you're looking for. Okay, the last one is fibrocartilage. This one's less solid or firm compared to hyaline. It's more compressible, and it is serving as this sort of intermediary or this linker between hyaline and dense regular connective tissue in the body. You can appreciate in this picture the lacunae and the chondrocyte in the middle, lacunae, chondrocyte in the middle. A lot of that you'll be identifying in lab as well. So that's the crossover. You find this between the intervertebral discs in the vertebral column or the backbone. You find this in the pubic symphysis, or where the two pelvic sides of the pelvis come together, the two pelvic girdles. Menisci in the knees, like a torn meniscus, is actually fibrocartilage. And you can really appreciate in this picture the wavy parallel lines from like upper left to lower right as they kind of stack together. So it's compressible and it's strong, but can it tear? Yeah, just like dense, regular connective tissue, fibrocartilage, meniscal tissue in the knees are very prone to tearing. And many of you that have torn an ACL, you're like, well, yeah, I did that, and I blew out my meniscus too. It's like, way to go, 120%, nicely done, okay? So that's why. Another question, I don't know. I don't know why it's oriented like that. Why wouldn't it be irregular? It would be stronger. Maybe it takes more biological energy, and so we kind of take shortcuts at some level. How do you tell if it's dense regular connective tissue or if it's fibrocartilage? The dense regular connective tissue is less wavy. It's more parallel lines. Do you see the difference there? And there's no lacunae. So here you have lacunae that house the chondrocyte, these open spaces, and then there's like a cell in the middle of it. Does that help? It's a good question. What's bone? So it's connective tissue, but it's also very much like cartilage. It's actually the final form developmentally after you're building a cartilage template. And we'll talk about ossification, both types of ossification when we get to the skeletal system. But we have two main types of bone. We've got what we call cancellous bone, or we actually have osseous bone. Now, there's numerous names for both. Cancellous bone is called cancellous, trabeculated, or spongy bone. It looks like a sponge. Cancellous, trabeculated, or spongy bone. Osseous is referred to as cortical or compact. Why are there three names, Dr. Keller, for the same stinking thing? It's a great question. Because different people study different aspects of the bone. They publish in the literature, and they call it their own thing. Okay? So cancellous bone has trabeculated structures in it. So that investigator was looking at the trabeculae, and it has a spongy-like appearance. So that's where it got its name. Osseous, it's more solid. It's like the outer surface of the skull. It's very compact. There's no open space. That's kind of where it gets its descriptors from. But both of these types of bone have what we refer to as hydroxyapatite. This is what's known as bone mineral. Hydroxyapatite is bone mineral. It's a natural occurring mineral form of calcium apatite, or a calcium deposit. And this is a cross-section through bone here, and you're looking at a structure. The way bone is organized is if you took the old number 2 pencils and you put a bundle of them together in a rubber band, and then you looked at them on end, and you could see all of the lead down the middle. That's kind of what you're looking at here, is the tip of the pencil, which is the central canal, and you're seeing all the different pencil structures. Some of them are fatter than others. Some of them are skinnier than others. That's why this one is wider, this one is thinner. Does that make sense? So that's kind of the orientation of this bone. And the main blood supply goes down that central canal. So I hinted at this in Monday's lecture, that the diffusion distance of oxygen is about what in tissue? Do you guys remember? 120 micrometers, pretty close. It's about twice that, about 250. Okay, but you're in the right scale. Okay, so 250 microns. And so does it surprise you as bone grows that the distance between the central canals is never further than about 500 microns, 400 to 500 microns? Because right here in the middle, these bone cells are getting oxygen nutrition from this one or this one. And if you get too big without building a blood vessel, then you're going to have necrotic bone. So that feature of the diffusion distance in tissue helps to build out our skin, helps to build out our bone, helps to build out how certain structures are the way they are. That principle of oxygen diffusing into tissue. Kind of cool, I think. Now, you'll see in lab, I mean, there's connections between the blood vessel, the central canal, and the lacunae. All these smaller dots are lacunae, and those connections are canaliculi. So you'll get into a lot of those more identifications in lab. But I think I kind of nerd out a little bit on this sort of orientation or architecture of bone and the why behind it. Okay, here's cancellous bone, trabeculated bone, spongy bone. The T, and we've seen this picture before on Monday, but the T is labeling the trabecula. This is hard bone. But there are little islands, and all in between all of that is adipose tissue that has like an open meshwork. It's a loose connective tissue. So it has this open sponge-like appearance, hence spongy bone, trabeculated bone, affectionately referred to as cancellous bone. So trabeculae is Latin for small beam. So you've got a beam here, you've got a beam here, or these little islands of bone or beams of bone, and in between them you pack it with marrow and adipocytes. This is a cool prep in lab, and I want to explain it. This is a decalcified bone. So you're actually seeing everything happening on this one slide, which is a pretty cool slide. You've got compact bone here. See how this compact bone is solid bone, right? It's not cancellous. It's not like this. It's all of these trabecula-like structures in compact bone. Here's an osteoblast, which is a bone building cell. Here's an osteoclast, which is a bone breaking down cell or a bone remodeling cell. And here's an osteocyte sitting in a lacunae, its little cave. So what's happening is these cells are laying down new bone as it moves to the upper right. These cells are chewing away this bone as it moves to the upper right. Isn't that interesting? So the bone's remodeling. We'll come back to this later in the semester. But right now, what I want to show you is this little picture, or video, excuse me, on bone remodeling that should help give you some insight as to what this looks like.
Skeleton changes across the human lifespan. This is characterized predominantly by bone formation and growth throughout childhood, followed by a gradual loss of bone density that begins with early adulthood that can accelerate significantly in older adults. The density of bone is modulated by a group of cells including osteoclasts, which are multinucleated cells that resorb bone, and osteoblasts, which refill the resorption cavities created by osteoclasts. Osteoclasts anchor themselves to the surface of bone. This creates a microenvironment underneath the cell, which is referred to as the sealed zone. Within this zone, the osteoclasts create an acidic environment that dissolves the bone's mineral content. Once the mineral content of the bone has been dissolved, enzymes released from osteoclasts remove the remaining collagenous bone matrix to complete the process of resorption. Following resorption, osteoblasts move into the resorption space and start to produce and deposit organic matrix called osteoid. Osteoid, a substance made predominantly of collagen, forms a scaffold in which minerals including calcium and phosphate begin to crystallize. Some active osteoblasts become trapped within the matrix they secrete and thereby become osteocytes. Other osteoblasts will undergo apoptosis or will revert back to lining cells which cover the surface of bone. This cycle of bone resorption and formation is referred to as remodeling. There is also a process where bone formation by osteoblasts occur without prior bone resorption by osteoclasts.
Okay, we're going to stop it there. I don't want to get too far ahead of ourselves. I'm just trying to give you a little bit of a peek as to how this stuff fits together. You saw terms in there like collagen scaffold, right? That's the extracellular matrix, the primary protein in the extracellular matrix. What's the secondary protein that's most commonly found in the extracellular matrix, other than collagen? Elastin. Well done. That forms a framework. It's like the old style jungle gyms in the playground. And then the kids will crawl on those bars. That's the cells as they migrate. And that video is trying to give you that picture of why this extracellular matrix is so important. Why do we care so much about loose connective tissue, dense or regular or irregular, or cartilage, bone? All of these connective tissues create an environment that the cells live in. That's what makes up your tissues. And the proteoglycan content, right? A sugar-coated protein attracts a sodium molecule that then attracts water, and that forms a gel, and that's why your tissues are hydrated tissues. You'll be doing some dissections in lab. You're going to cut into brain, right? You'll cut into eyeballs, and they're squishy. I saw some faces. That's where that water comes from. 50 to 75% water. That's what we are, right? Depending upon our hydration levels. 50 to 75%. Okay. The last type of connective tissue, then we're going to get into muscle, and then we'll be done. Blood. Red blood cells and white blood cells are in a fluid matrix. Someone appropriately said a function of connective tissue was transportation, right? Blood transports things. This tells me you guys are understanding what we're talking about. You're not just memorizing stuff. That's what I want, because you're going to move on in your careers, and you're going to use this semester and next semester as a foundational class. That's why you're taking this class, right? How many of you are going into the health professions? Yeah, that's what I thought. So this is a picture, a slide of blood. You're going to get more in detail in 202 because we talked about the cardiovascular section in 202. But because it's a connective tissue, we actually discuss it in 201. And you'll look at slides probably in lab on blood. All of these red sphere structures are red blood cells, erythrocytes. Every now and then you're seeing a weird-looking cell, which is a leukocyte, also referred to as a white blood cell or a WBC. So the matrix is only really visible when you have a clot. Otherwise, it's in liquid form. And the plasma that it floats in is a protein that's not collagen and it's not elastin. It's mostly albumin. Albumin is the protein that makes up the extracellular environment of this particular connective tissue. And the liver is what manufactures your albumin. The liver makes the albumin for your body. Okay? One of the many important things that the liver does. Okay, let's look at some histology on muscles and nerves and talk a little bit about what they do as we kind of wrap things up. And then I've got a little bit of a practice for you at the end. So muscles, their function is to contract, to shorten. We will come back to this in the muscle physiology section. And this is where the class will kind of come together with lab momentarily. And then we will move away because you're going to be talking about muscles and memorizing muscles and insertions and action and where they are for a few weeks. And we're going to have 3 lectures on muscle physiology, like a week and a half. Make sense? But we'll come back to the purpose of the muscle. You're going to memorize and understand where they are and what their identity and what their names are in lab. In here, you're going to talk about how they work. So it's mostly the structure, architecture in lab, and it's mostly the physiology in how they work in lecture. But right now, we're going to talk a little bit about both. So they function to shorten. They have they're a cell. A muscle cell is a cell, but it's a specialized cell. It's a tissue that is cell-rich and very well vascularized. But these are elongated cells. So they have a slightly different shape. In fact, they're really, really long cells. And when they shorten, they contract inwards. So they have cytoskeleton like actin and other cytoskeletal proteins like myosin and tropomyosin that regulate whether they shorten and how much they shorten. So we have three main types of muscle: skeletal, cardiac, and smooth. Skeletal and cardiac are more similar to each other, and smooth is kind of the different one. So skeletal muscle, we're going to look at each of them individually. Skeletal muscle is shown on this slide. It's typically under voluntary control. Some may argue, well, in the case of a reflex, it's under involuntary control. That would be an example of an exception. It's attached to bones via tendons. We already kind of talked about that. These elongated, multinucleated cells have this striated appearance. And the striations that you can appreciate on this right image are because of the cytoskeletal proteins and how they're arranged, how they're organized. So they're highly organized cytoskeletal proteins known as myofilaments, and that's what gives you this giveaway from an identification standpoint. Is you've got multinucleated, striated muscle in a parallel fashion. Those are fibers that you're looking at. So cardiac muscle, very, very similar, but it's under involuntary control. Like, you're not sitting here thinking about your heart rate. You're not thinking about like beat, 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 beat. If I call on you to ask a question, you're not like, oh boy, beat, 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 right? It just naturally does it on its own, involuntarily. So that's a big difference between cardiac and skeletal, but they look very, very similar because they're both striated. This one, though, is uninucleated versus multinucleated. So many, many nuclei in the cell of a skeletal muscle, only one nuclei. That's what uni means, like unicycle. Unibrow, right? I know that one. Striated cells, they're very, very similar in their striations appearance, just like skeletal muscle, because they have the same cytoskeletal filaments. But they have a unique structure, which is called an intercalated disc. And that's these dark lines that connect one cell to another. We talked about this. And this is labeling these intercalated discs, which actually electrically communicate one cell to the next, because the heart needs to beat in sequence. And so the way the heart is developed embryologically is it develops as a tube, like a blood vessel. And then it actually folds like a U, and then it presses together, and then it pinches off 4 different chambers. That's how it's made embryologically. And so if you take the tube and you'll look at cardiac muscle contraction in 202, it's like squeezing a toothpaste tube. Best results, squeeze from the bottom, right? It says on the back of the container. You're like, you've actually read that? It's like, yeah, that's how I am. So if you squeeze here, and then squeeze here, and then squeeze here, and squeeze here, the fluid goes that way. And that's what the heart does. So you need these intercalated discs to communicate. Squeeze here, squeeze here, squeeze here. You don't want to squeeze here and then squeeze here, right? That's a competing interest. So you have this electrical connection of intercalated discs in cardiac only. That's a unique feature to cardiac muscle. Smooth muscle. Now, smooth muscle, I told you, typically gets misrepresented as dense, regular connective tissue. Remember that? I can go back to that picture in a second. But this has far more nuclei. So you can see how it's organized in parallel. There's no lacunae, because one of your classmates was asking, well, how do you know this isn't cartilage, right? There's no lacunae. Let me go back and show you the difference real quick. And this stuff will help with lab, not just with lecture. So this is the one. Do you see how few nuclei there are here versus the smooth muscle picture? Make sense? So these are slowly contracting muscles, very different than skeletal and cardiac. They don't have striations. They're just organized in this direction, but there's no striations because the myofilaments inside the cell are organized in a more random pattern. And we'll look at that later when we get to the muscle physiology section. But because of that, they're more of a fusiform. A fusiform is a word that means it's kind of wide in the middle, tapered on the ends. Wide in the middle, tapered on the ends. Kind of like a yam or a sweet potato. Okay? No visible striations, slow contractions, involuntary control. So you eat lunch, you had a snack before you came. You want to move the food through the alimentary canal. You want to break it down chemically, which is digestion. Then you want to be able to absorb the nutrition across your GI tract into the bloodstream. If you contract your GI tract, boom, 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 you eat and then you're like, right? It goes right through. But if you contract super slow, it moves slowly, it gives time for you to absorb the nutrition. So, I know it sounds weird, but you have a meal, right? A patient has, a kid swallows a nickel. You're like, don't worry, it's going to pass. Well, it may take a day or two before it shows up in the toilet because these are slow contractions to move this. If there's irritation, it moves quickly, and that's in the form of what we talked about earlier, diarrhea, right? So very different strategy, very different muscle because it's a very different purpose. Okay, the last tissue type is nervous tissue. We'll look at the nervous section towards the end of the semester, and then we'll get into special senses, and that'll kind of track with lab. We'll finish up with special senses here, and lab will finish with special senses like vision, smell, or olfaction, hearing, equilibrium, balance, those kinds of things. But what we talk about with respect to nervous tissue is two main types of tissue. We either have the neuron or we have the supporting cells known as glial cells. Now, the glial tissue outnumbers the neurons 9-1 in nervous tissue. There's far more supporting cells or glial cells than neurons or nerves. There's lots of nerves, but there's 9 times more glial cells. If you're in the central nervous system, I'll give you a clinical correlation. You're in the central nervous system, and you have a patient that unfortunately wrestles with a central nervous system cancer that's very aggressive. It targets a specific cell type. It spreads very rapidly. The prognosis is typically not great. This is what Senator John McCain passed away, if you remember that. And this is a glioblastoma, where it's these cells that mutate, which are all these smaller nuclei. The big giant one in the center stage is the neuron. But the glial cells outnumber the neurons 9-1. Neurons are very important. We're going to spend a lot more time on neurons, but I'm trying to give credit to the glial cells, which are really important. We'll talk a little bit about them. But the main character of nervous tissue is the neuron. It sends the signal. It sends the electricity. It's the wire that you plug in to charge your iPhone, right? That's the neuron, to use the analogy. Sends and receives information. We have dendrites, we have soma, and we have the axon. The dendrites are what receive the image. The soma is another word for cell body. And then the axon is where you send the signal down to the next neuron. These glial cells are much, much smaller, as you can see. There's different types that we'll get into. We won't get into it today. But I just want you to, remember when we're foreshadowing to the future, and then we'll flash back to this lecture when we get to the nervous physiology section. When we start looking at the many different types of glial cells. There's 4 different types in central nervous system. There's 2 different types in the periphery. Okay? So the word dendrite in the Greek means tree. So dendrites have sort of this branching, it looks like aspen tree in the winters when they drop their leaves. That's what the dendrites look like as you get into lab. Questions? Any questions? Okay, I have a practice quiz for you to do now. We're going to do it together, okay? You ready? So, what is this? Between the arrows. What is this? Just think it through. Don't shout it out yet. Just think it through, and then I'll ask for a volunteer. 4 options. The cell between the arrows. Any volunteers? What's that? C, simple squamous. I would agree. Very, very well done. Okay, what about this one? What is it? Is it dense? It's not even an option, Keller. I know, but what does it look like? Does it look dense or does it look open? All right, any volunteers? What's that? Elastic? It's a decent guess because you're seeing the elastin, which is these black squiggly lines. But elastic cartilage, you're going to see lacunae, and you're going to see a chondrocyte in the middle of it, okay? In the back. Yep. E, areolar connective tissue. Well done. Areolar connective tissue. It's open, it's airy, that's right. That's kind of why I was saying that in the beginning, try to give you a little clue. Okay, how about this one? I'll ask for a volunteer. If you raised your hand before and I didn't call on you, then you're up first, if you want. A. It's columnar. It's columnar, but it's not actually stratified. So what is it? E, it's pseudostratified epithelium. It'd be better for me to say pseudostratified columnar epithelium, right? Ciliated pseudostratified columnar epithelium. That's kind of a bad test question, but this is pseudostratified ciliated columnar epithelium. D and E? Yeah, they're very different because simple columnar is going to look like. That's a good question, though. Simple columnar is gonna have this. Like all the nuclei are going to be at the same level. Pseudostratified is this one where you've got. Does that make sense? So the nuclei are kind of at these different levels. This is a hard one. That's why I keep bringing it up. But once you get it and you see it, you'll never forget it. You'll be like, it kind of becomes your favorite because it's like very unique in a weird, nerdy kind of way, right?
Is the cilia only a feature of pseudostratified, or is it also on the columnar?
Great question. Is the cilia a feature that's only on pseudostratified, or is it on the simple columnar? It is only on pseudostratified. And that's why we get lazy and we say pseudostratified epithelium, because it's always ciliated. Okay? Great question. There's another question. Same question. Okay. Great minds think alike. Okay. What's this one? Oh, where are you most likely to see this tissue? So first you got to identify it. Now you got to tell me where it is. So let me ask you again, which one is this in the maroon? Which one do you Yeah, it's this one right here. So where are we going to find it? A, gastrointestinal lining. Well done. Lots of absorption happening there. Make sense? So what you're starting to do is put together structure and function, right? The way it looks, the shape has a purpose, and then it gives you clues as to what the physiology is. Okay, how about this one? What is it? It is C, reticular connective tissue. Well done. What is this one? Any guesses? A, stratified columnar. It is. It's not ciliated, so it's not pseudostratified. That's not even an option. But it's definitely not just one row of cells. Okay, how about this one? The small black fibers on this slide, what are they? What are they? Elastin. Okay, good. Good. You guys are listening. Okay, bonus. What is the tissue? What kind of tissue do you think it is? Reticular, it's open like a reticular, but it's not reticular. It's another open kind of, what's that? Areolar. I know, these are hard. That's why I'm trying to reinforce them so you kind of have it for lab and lecture. Okay, we're getting close. We're getting close, folks. We'll finish a few minutes early. What's the major function of this tissue? Major function of this tissue. First, you kind of have to ID it to tell me what the function is, right? Right here, do you want to guess? Yeah, did you have your hand up? Sorry. E, storage of calcium phosphate crystals. It doesn't have the lacunae, and there's no osteon, so it's not bone. A, movement of digestive food. You're thinking it's smooth muscle? It's not smooth muscle. It's a good guess, but it's something that looks like smooth muscle. C, connects bones with bones. It's dense, regular connective tissue that's more highly nucleated than the one I gave you an example of, right? Yeah, I know. But I'm trying to show you the variations here. Okay. How do you know it's not smooth muscle, Keller? Right? That would be a tough one, but you probably could argue on the exam A, because it's very difficult to discern if this is smooth muscle or if it's dense, regular connective tissue. That's why I went with A, because I thought it was smooth muscle. That would be an example of how to argue a question. Does that make sense? Like a diplomatic, scientific way to challenge a test question. And we'll have that dialogue. Okay, good. I try to eliminate vagueness on the exams, but I'm human. Sometimes I make a mistake. Okay, what is this one? Transitional epithelium. Wow, that was your question, wasn't it? Okay. All right. Couple more. What would you call this mature cell in this tissue? What was it? Somebody say B? Yeah, B, adipocyte. An immature cell in this tissue would be called what? You see lacunae? This is fibrocartilage. So it would be a fibroblast, A. What is this tissue? What is it? Not even asking what it does. Do you see striations? No striations. Smooth muscle, C. What is the arrow pointing to? Remember the Greek? Huh? Dendrite. Well done. All right, how about this one? The arrows kind of give it away if you focus on the arrows. Cardiac muscle. Well done. What about this one? Skeletal muscle, A. Okay, you guys are getting better. What is this one? Smooth muscle. What is this that the arrow is pointing to? It's the nucleus of the neuron. Neuron's not even an option, so you got to choose something else. How about this one? Stratified, because there's two layers, I agree. Cuboidal, because you can kind of appreciate the squares, right? I mean, they're not perfect squares. I mean, we're talking about biology here, folks, right? Okay. Questions? Comments? Question here, and then I'm going to pause the recording.